Proteins have 4 stages of structure, the first one being the protein's primary structure. This is the unique sequence of amino acids in a polypeptide chain and is the fundamental structure of a protein. Next is the protein's secondary structure, which is formed by local folding of the polypeptides in specific places. The helix and the pleated sheet structures are the most prevalent. The helix structure is present in both forms. Hydrogen bonds keep the helix of the protein's secondary structure in place. We then have the protein's tertiary structure, which is a unique three-dimensional structure of a polypeptide. Chemical interactions on the polypeptide chain contribute to the structure, whereas the interactions among R groups are primarily responsible for a protein's complicated three-dimensional structure. Finally, we have the quaternary structure. Unlike the previous three structures that only have one polypeptide chain, multiple polypeptide chains, also known as subunits, come together and give the protein its quaternary structure. Because of carbohydrates' many roles in all kinds of life, they account for the majority of biological stuff on Earth. Carbohydrates are used as energy reserves, fuels and metabolic intermediates. Carbon, hydrogen and oxygen make up the carbohydrate structure. Carbohydrates have the same structure and function as monosaccharides. Monosaccharides are simple sugars and are joined by the glycolytic bonds, which arise as a result of a dehydration process, forming disaccharides and polysaccharides with the loss of a water molecule for each bond formed. Biological fatty acids, which belong to the carboxylic acid class, are made up of a hydrocarbon chain with one carboxyl group at the end. An acyl group is a carboxylic acid fragment that does not contain the hydroxyl group. This acid group has normally lost the hydrogen ion to produce a negatively charged carboxylate group under the physiological circumstances in water. Most biological fatty acids have an even number of carbon atoms, although relatively small amounts of odd number fatty acids do occur in some organisms.
Formation and breakdown of polymers. Polysaccharides are polymers made up of multiple monosaccharide molecules joined together. Glycosidic linkages generated through condensation processes and connected the monosaccharides. Polysaccharides are insoluble because they are big molecules. They are useful for storage because of this property. Polysaccharides are broken down into disaccharides or monosaccharides when they are hydrolyzed. Hydrolysis is a procedure that breaks down polymers into monomers by using a water molecule. When complex hydrates are hydrolyzed, monosaccharides are released. Proteins are natural condensation polymers made up of thousands of amino acid units joined together. The amino group and the carboxyl group are both functional groups in amino acid molecules. Lipid produce a special sort of polymer that is a crucial component of cell membranes and hormones. Some lipids, such as cholesterol, can create polymer-like structures with another type of fat molecule. Structure of enzymes and how it's linked to their function. Enzymes are enzymes which are Structure of enzymes and how it's linked to their function. Enzymes are enzymes which are proteins that catalyze processes. They all have primary, secondary and tertiary structure, and some have quaternary structure if they have more than one polypeptide chain. Because of the varied properties in the amino acids, primary structure is what determines overall structure, such as if there are acidic or basic within the polypeptide sequence, hydrogen bonding between the N equals H and C equals bonds of the protein backbone forms secondary structure, such as alpha helices and beta sheets, bonding between the R groups of amino acid residues in the same polypeptide forms tertiary structure, which gives the enzyme its overall 3D structure. The same sorts of bonding occur between residues from various polypeptide chains in the quaternary structure. Sensitivity is provided by enzymes' complementary structures to their substrate. They cause the substrate to be strained, causing it to enter transition state, which gives catalytic characteristics. By lowering the activation energy, the characteristics of the residues in the active site as well as how they interact with the substrate account for this. The induced fit model is what it's called. They are no longer complementary to the enzyme's active site and diffuse from the site after product is generated. Activation energy. The lowest quantity of energy required for a reaction to occur is known as activation energy. If less energy is required, reactions can occur at lower temperatures than would be required without the use of an enzyme. Some or all of the chemical bonds in the reactants must be broken for the reaction to occur, allowing new bonds, those of the products, to form. The molecule must be twisted, deformed or bent into an unstable condition, termed the transition state, in order for the bonds to be able to break. The transition state is a high energy state that requires the addition of energy, the activation energy, for the molecule to achieve it. Reactant molecules do not stay in the transition state for long since it is unstable, and they swiftly move on to the next step of the chemical reaction. Lock and key method. When a substrate molecule collides with an enzyme with a complementary active site shape, the substrate will fit into the active site, forming an enzyme-substrate complex. The enzyme will catalyze the reaction, and the products will form an enzyme-product complex with the enzyme. A reverse reaction can be catalyzed by an enzyme, according to this concept. The lock and key concept depicts an enzyme as a stiff structure and only able to link to substrates that match the active site perfectly. Emil Fischer proposed the lock and key analogy in 1894 to illustrate the unique action of an enzyme with a single substrate. Induced fit. Induced fit hypothesis is a more contemporary concept that is supported by evidence and largely recognised as understanding how enzymes act. It argues that the structure of active sites isn't exactly complementary, but that when a certain substrate is present, it is shape to become complementary. If the composition of a substrate molecule collides with an enzyme, the shape of the enzyme's active site changes, allowing the substrate to fit into it for an enzyme-substrate complex to form. The reaction is then catalyzed, resulting in the formation of an enzyme-product complex. The induced fit model depicts that the enzyme structure as more malleable, complementing the substrate only after it has been bound. Factors affecting enzyme activity. Temperature. The kinetic energy of molecules increases as the temperature rises. As a result, the molecules move faster and collide with one another more frequently. This indicates that in an enzyme catalyzed process, the enzyme and substrate molecules come together more frequently in a given amount of time. More effective collisions result in the formation of more enzyme-substrate complexes, increasing the pace of reaction. The optimal temperature for several enzymes in the human body is above 40 Celsius.